This video is about the birds and the bees, both figuratively and literally. In this overview of the evolution of sex, I'll be focusing on pollinators because they provide critical ecosystem services and have rich coevolutionary histories with plants. Oh, and pollinators aren't all bees and butterflies. We'll also be talking about other insects, as well as birds, bats, and even a lizard. Why are these common bush hoppers engaging in sexual reproduction? As humans, we might ask if they're doing it for pleasure, but sex predates the evolution of the animal nervous system, so pleasure is certainly not the ultimate causation for this behavior, for this genetic process. At first glance, there doesn't even seem to be a strong reason for sex at all. Sexual reproduction is costly. There is an immediate 50% fitness cost to sexual reproduction compared to parthenogenesis, which is a form of asexual reproduction carried out by females. Beyond this initial cost of fitness, sexual reproduction also requires organisms to spend time searching for mates. Of course, it's often difficult to find a mate, and there's a possibility that the mate is infertile or the two partners are otherwise incompatible. The act of copulation itself can pose a risk of physical harm or disease transmission, and at a genetic level, we may lose particularly useful combinations of genes that, independently, just don't have the same effect on phenotype or the fitness of that individual in a particular environment. Despite the costs, there's an incredible diversity of sexually reproducing species, especially among plants, animals, and other higher eukaryotes. We call this the paradox of sexual reproduction. Even after factoring in all of the possible fitness costs, there must be some net benefit. Otherwise, sexual reproduction should be an extremely rare phenomenon. It turns out that sexual reproduction is particularly advantageous in unstable or changing environments, right? That describes most environments. This is encapsulated by the Red Queen hypothesis. The Red Queen was a character in Lewis Carroll's Through the Looking Glass, although she's often confused with the Queen of Hearts from the original Alice's Adventures in Wonderland. The Red Queen hypothesis states that continuous adaptation is needed to avoid extinction in a changing environment, and its name comes from the quote, "Now, here you see, it takes all the running you can do to keep in the same place." This suggests that the greatest benefit of sexual reproduction comes from meiotic recombination. Meiosis occurs during gametogenesis, and this process involves genetic recombination and the independent assortment of homologous chromosomes into separate gametes. Recombination increases variation among genotypes and lowers selective interference, which essentially means that alleles are separated from an individual's genotype or genomic background, so selective forces can act on the level of the gene. But it's also worth mentioning that selective interference can still be high in sexually reproducing populations because some chromosomal regions have relatively low recombination rates. Okay. Another major benefit of sexual reproduction is outcrossing, which can ameliorate inbreeding depression or help populations with otherwise limited variation adapt to a changing environment. These are all major benefits. So despite the obvious costs, Knapweed fritillaries and many other eukaryotes continue to reproduce sexually, although there are, of course, a few groups that have also found asexual reproduction to be adaptive under certain circumstances as well. Let's take a look at a hypothetical comparison between parthenogenetic and sexually reproducing females. In both cases, each diploid female has one pair of chromosomes and will only produce 2 offspring. For our parthenogenetic female, her 2 female offspring are nearly perfect clones, assuming the mutation rate is 0. The coefficient of relatedness between mother and offspring is 1. They share 100% of their alleles. For our sexually reproducing female, she'll need to mate with an unrelated male before she can have any offspring. Her offspring will be 1 daughter and 1 son. An even mix of male and female offspring like this is a common assumption to make, but there are always exceptions. Now, for sexual reproduction, the coefficient of relatedness between parent and offspring is 0.5, because through meiosis, only one of her 2 homologous chromosomes can be passed to each offspring. This poses an immediate 50% fitness cost compared to parthenogenetic reproduction. But because homologous chromosomes contain different alleles of the same genes, recombination unlinks genes that are further apart from each other and allows for different combinations of alleles to be passed to offspring. The coefficient of relatedness between mother and offspring will still be 0.5. Continuing to the next generation, while parthenogenetic females can continue to reproduce clonally, sexually reproducing females must find a partner before reproducing at the same 50% cost of fitness. Also, because males are incapable of producing offspring on their own, this effectively cuts the overall population growth rate in half, and this is the twofold cost of males. But males do pass on their genes if they can find a mate, so this twofold cost can also refer to the relative cheapness of males, as they're less efficient at converting their resources into actual offspring. Anisogamy, or gamete size dimorphism, occurs in plants, animals, and several other eukaryotic groups. We define females as the sex that might have, or does, produce relatively large, and often non-motile or non-moving, gametes. Males are defined as the sex that might, or does, produce relatively small but numerous, and often motile gametes. This size versus number dichotomy is obviously an energetic or resource-limited constraint that early on may have been subject to disruptive selection. Anisogamy itself contributes to the sexual dimorphism that we see in many species. Sexual dimorphism is also associated with many secondary sex characteristics, which are sexual traits not associated with the genitals or reproductive system itself, although all of these are influenced by sexual selection. Look at this female fuzzbuzz. Bumblebee. Female and male bumblebees differ in phenotype, in their primary and secondary sex characteristics. Female and male individuals experience different magnitudes of selective pressures. Bateman's principle observes that there is higher variance in the reproductive success of males, and so sexual selection acts more strongly on males than on females. Key processes include female choice or female preference, male-on-male combat, alternative mating strategies like sneaking and female mimicry, as well as post-copulatory mechanisms like sperm competition. All of these selective pressures can affect male phenotype, like when females of some species of paper wasps prefer males with black color patterns on their faces. Of course, males are not always under higher selective pressures. When females are under a higher pressure, this is called a sex role reversal. Female preference can appear through a variety of mechanisms. Direct benefits, like nuptial gifts or parental care, effectively increase female fitness, and, as such, are selected for. In the absence of superfluous, maybe, gifts, good genes can also be selected for if the physical observable traits of a male are an honest signal of fitness, that the resulting offspring are more fit, which contributes to the overall fitness of the parental female, of course. On the other hand, female preference may be due to perceptual biases, which are responses to stimuli that originally evolved within some other context. Perceptual biases include sensory traps and the exploitation of latent preferences. Sensory drive is an evolutionary concept that integrates evolutionary genetics, like genetic correlations and pleiotropic effects, with sensory exploitation, traps, and biases. So we can think of it as how a variable environment influences communication. Context, as well as the sensory capabilities of the receiver. But sometimes there can be positive feedback loops in the selection of certain traits. Drosophila fruit flies, which pollinate certain orchids in Central and South America, have males that produce giant sperm, and these giant sperm are a type of elaborate ornamentation that evolved by Fisherian or runaway selection. So, how are females and males differentiated, genetically or developmentally? There are a few major mechanisms of sex determination. Let's start with perhaps the most familiar one, the XY determination system. In this system, females are the homogametic sex, while males are the heterogametic sex. This male hoverfly, for example, has an XY chromosome complement. It is the heterogametic sex. The ZW sex determination system is kind of the opposite of the XY system. Here, it's females that are the heterogametic sex and males that are homogametic. This female New Zealand bellbird has a ZW chromosome complement. The XO determination system is related, but a bit more complex. In this case, there are homogametic, typically XX females, but functionally heterogametic males who have either lost a Y chromosome or, by comparison, lack that second X. This female goldenrod spider, which, I'm sorry, is not a pollinator itself, but a common predator of insect pollinators, has an XX chromosome complement. Males of its species only have a single X chromosome and nothing else. Next, we have environmental sex determination systems, which come about through a diversity of underlying genetic and physiological mechanisms. There are various deciding factors, including temperature, and these factors can produce different patterns, depending. This day gecko, for example, developed as a female due to relatively high temperatures. Day geckos and other lizards can be particularly important pollinators, especially in island ecosystems that might lack other, more conventional pollinator species. Finally, we have haplodiploid sex determination, or haplodiploidy for short. Here, females are diploid, developing from fertilized eggs, and males are haploid, developing from unfertilized eggs. At least, that's the simple explanation. It's usually more complicated than that. For example, this female honeybee is diploid or heterozygous at one or more complementary sex determiner loci. Males then are hemizygous or homozygous for CSD. Haplodiploid species can have very female-biased sex ratios. This means there are many more females than there are males. This can be an adaptive response to local resource competition, LRC, or local mate competition, LMC. In honeybees and other eusocial insects, differences in relatedness can create conflict between workers and their queen. In eusocial bees, workers share 75% of genes with sisters, but only 25% with brothers. There are a lot of other complex phenomena associated with haplodiploidy as well. Anyway, we would normally expect to see a 50-50 sex ratio of 1 female per 1 male, which is the evolutionarily stable strategy, as conveyed by Fisher's principle. Fisher's principle states that the rarer sex would be expected to have a mating advantage. So, analogous to negative frequency dependent selection, natural selective pressures favor equilibrium. Bee flies, like this fuzzy little guy here, have a nearly perfect 1-to-1 sex ratio. In case you didn't know, bee fly larvae are strange parasites of bees, but the adults are generalist pollinators. There are many, many more ecoevolutionary processes related to sexual reproduction, like the coevolution of plants and pollinators, reproductive interference, which includes heterospecific rivalry, heterospecific mating, misdirected courtship, erroneous female choice and hybridization, as well as kin groups and sociality. But before we go, don't forget that bats work the night shift. I haven't had a chance to mention this specific group of pollinators yet, but they're very important for the pollination of mango, banana, and other plants. Alright, thank you for taking the time to watch this video. If you have any questions, feel free to reach out to me.